Hi, I'm Lucia Gambuza with Modern Retina, joined by Eric Schlumpf, President and CEO of Stuart Therapeutics. Eric presented at OIS Retina 2026 during the Innovation Showcase on the company's collagen mimetic peptide platform and its application in retinitis pigmentosa, the first time the company has presented RP as an indication. Thank you for joining me today.
Pleasure to be here. Thank you for having me.
Let's jump right in. What is the mechanism behind the collagen mimetic peptide platform? What made retinitis pigmentosa the next indication to advance on it?
Yeah, thanks for asking. The mechanism behind the collagen mimetic peptide is a direct reparative of helical collagen structures in the body that are damaged in disease processes. What we find is that these helical collagen structures not only help maintain the physical support nature of collagen for cells in the microenvironment, but they also provide a cell signal ligand binding site for important inflammatory modulators and cell growth and proliferation signals. The effect of the mechanism of action is that the peptide binds to these damaged locations, restores the structural component of collagen, and flips the cell signaling switch towards inflammatory modulation and improvement in the cellular environment, which happens quite quickly. So we've seen the effect of this in our ocular surface disease programs in the anterior segment, and we've done some testing in a couple of different animal models for retinal disease in general, and we looked specifically at retinitis pigmentosa as an option in our retinal program more recently.
What is RD12 model? What was the study designed to answer, and what did the structural and functional measures show?
Yeah, RD12 is a knockout mouse model for Leber's congenital amaurosa. And it's an aggressive, it's designed to be an aggressive decline of the retina overall. In particular, total loss of retinal thickness happens very, very quickly. And we did that as an initial test because it was an aggressive model. We wanted to see what effect the peptides would have on this. And what we got out of it was a couple of different measurements that were important. One was retained retinal thickness was improved with statistical significance by the use of the peptide as a therapeutic. We also did some histology that showed that the cells in the posterior retina were improved dramatically with treatment compared to controls. And then finally, we did some very early visual function tests in between the intravitreal injections, which was the mode of administration. And what we saw in each of the intervals between injections was an improvement trend on visual function with the application of the peptide when compared to controls. So that original model that we presented at OIS Retina gave us some signals that suggested that this was going to be a potentially an important therapy for retinal disease in general, and potentially in retinitis pigmentosa or other inherited retinal diseases. We've since done one additional model using an RD10 model that confirms the visual function improvement, and we're still gathering data from that set of tests, and we'll have more to report later in August.
What stage of RP is this program being designed for, if any? And what does the preclinical work suggest about that point of no return for RP?
Yeah, I think our point of view about the peptide is that as a reparative, we have the ability to help repair nerve cells. We've shown that in the work we've done in glaucoma and some of the things that we've seen in the corneal nerve bed. And the early work in retinal disease suggests the same for the nerves in the retina. So it is neuroprotective in nature. What we don't do is we don't grow new nerves. So if there has been a complete destruction, death of the nerve cells in the retina, we're not going to make new ones grow back. So we think it is an earlier stage therapeutic for retinal disease. It is non-gene specific. So if we get into inherited diseases with the therapeutic, it would be a non-gene specific therapeutic and could be used in conjunction with other therapies because it has anti-inflammatory properties and contributes to the overall health of the posterior retina.
That's a great follow-up to my next question. Where does gene-agnostic neuroprotective approach fit alongside gene replacement, optogenetics,
Right.
and cell-based therapies that are already generating multi-year clinical data?
Right. It's early days, and we're still learning as an industry about the effects of gene therapy and what's really going to be viable and what's not going to be viable. Our hope is that if a patient were to be diagnosed with an inherited retinal disease, for example, that this could be a standard therapy that could be used to delay or delay significantly onset or reduce the impact of the onset of that condition while the patient was preparing for or perhaps even underwent gene therapy. As it relates to cell therapy, we don't really have a position on that yet. However, what we do know about the peptide in our ocular surface work is that we're beneficial to endothelial cells, epithelial cells, and nerve cells. So if there was an RPE cell replacement, for example, the use of the peptide would support that and would create a healthier microenvironment through repair of the collagen. I think overall, a non-gene specific approach means a broader opportunity in the market for us, which we're excited about. And we're very hopeful that we can carry this through and bring something to the market that provides benefit to particularly younger patients that are diagnosed with these difficult conditions.
Absolutely. What endpoint is the RP program being built around for a first-in-human trial? What would you like to see?
Well, we're still knocking that around, honestly. We're in discussions about this. As I said, we did the first. We've done a number of in vitro studies on RPE cells, but our first in vivo test was the RD12 LCA model. The second was the RD10, which we're just wrapping up now. And I think we're going to sit down and we're going to narrow down gene types for inherited retinal disease, and we're going to narrow down endpoints, and that'll likely end up being in conjunction with conversations with FDA.
Absolutely. And how long would that trial need to run, if you know, to show an effect?
Yeah, I think that's going to be dependent upon the specific gene variants that we put in a trial because they operate, as you know, they operate at different rates. Some are very aggressive, others take years and years and years to develop. So I think we'll be looking for indications that allow us to showcase the reparative and healing capability of the peptide in a short enough trial that it's affordable and we can demonstrate a signal that leads to further development.
Absolutely. That's a good point to bring in the affordability of the trial
Yeah.
as well. This is my last question. What
Okay,
is
sure.
the single biggest unanswered question about using collagen repair to slow retinal degeneration?
I think the single biggest question that we have on our minds right now is the specific indications that we want to trial the drug in. I think that's something that we're gathering data for. Some of that's a clinical question, that's a regulatory question, that's also a competitive question, right? You want to understand a place that you can be successful in all three of those areas. So we're working that out now. That's the biggest question we need to get to.